A very good morning to Dr. Wee. We are from Group Hermo. Today we are going to present the poster presentation and the title is Mutual Interaction Between Arsenic and Biofilm in a Mining Impacted River. The abstract gold mining activities may cause arsenic pollution in Bluebird system, which is occur at the Inland River, Northwest Spain, where the concentration of arsenic in surface sediment will be found. Experiments were carried out in this river by moving some biofilm, colonized substrata from less arsenic polluted, which is upstream, to the more arsenic polluted in the mine area. It is to explore the effects of arsenic on benthic biofilm and their work on arsenic retention and speciation in the water sediment surface. Eutrophic conditions from 0.07 to 0.338 mg per liter total phosphorus were investigated in water in both sites, while sediment below than 600 mg per liter per kg were not considered as P-polluted. Dimethyl arsenate was found intracellular and a detoxification process by biofilm are suggested in the river. Our study provide new arguments to understand the contribution of microorganisms to the arsenic biogeochemistry in freshwater environments.
For the introduction, I'm going to introduce biofilm, arsenic and sediment. Microorganism is a form of major part in all living matter. The most of microorganisms living in the form of multicellular aggregates, which is commonly referred as a biofilm, such as bacteria, algae, fungi and protozoa. The role of biofilm in environments are maintaining and improving the ecological health of freshwater ecosystem. And in the most of the river, biofilms interact firstly with the dissolved substance such as pollutant, having a high capacity to accumulate heavy metals from the surrounding environment and being able to actively influence the desorption and transformation. Biofilms also a biotransformation site and chemical transfer to other aquatic organisms. For arsenic, in river system, arsenic is predominantly bound to sediment, which may contain high amount of this element, especially in mining area. According to the Sabitli and Kenny Burch 2002 and Rubinos in 2011, during the mining time, the concentrations of arsenic can be reached up to 100 or 1000 of milligram per kilogram in sediment. The role of sediment, it is act as a sink for pollutant that can subsequently be released upon change in environmental conditions. For the objective, our objective is to show the mutual interactions between arsenic and benthic biofilm in the mining impacted river. And the second objective is to study the contribution of benthic biofilms to arsenic biochemistry in freshwater environment. And the third one is to show the importance of biofilms as an indicator of the quality of freshwater ecosystem.
So I'm going to explain about the methodology. Water sampling, we will collect 1 liter water sample and the water sample will be filtered using GF/F glass microfiber filter. And the water sample will frozen until the analysis. So the last one is the GF/F filter will also kept for dry weight determination of river water suspended solid. For sediment sampling, we will collect the complex sediment sample and mix in situ in hermetic plastic container. And the sediment sample will centrifuge, freeze, dry and shift. For biofilm sampling, the samples will preserved in glass vials and will be kept in dark condition in the fridge for 1 month for the diatom analysis 3 months for bacterial density. And biofilm from the large substrata was scraped into the Falcon 50 ml tubes, and then some fresh sample were used to determine the concentration of total bioaccumulated arsenic and its species distribution in the different biofilm compartment, and other samples were freeze-dried.
For the result, based on the table, we can see that the river water, the total arsenic in the river water is low and it is not polluted. But based on the sediment taken from the river at the same site, there are a high value of sediment compared to the river water. The table 2 is about the benthic biofilm analysis. If we compare the two tables, there are a lot of difference in the value of arsenic concentration. The arsenic concentration by using the benthic biofilm has a much higher value. This is because the benthic biofilm is the arsenic density for this analysis. The bioaccumulated arsenic concentration in the upside were always higher than the area at the downside. The arsenic concentration in the biofilm exceed the easily extractable arsenic. Thus, it suggests that the arsenic accumulation over time is higher by using the biofilm. The arsenic was the predominant species studied in the river.
In conclusion, the mutual interaction between arsenic and benthic biofilm in the mining impacted river was shown, and it showed that the toxicant was mostly associated with sediment. Biofilm accumulated more at the site with higher arsenic concentration in sediment compared to site with low concentration of arsenic. Besides that, the presence of methylated arsenic species give an idea that there is a detoxification process occur within the biofilm. And next, the study provide information on contribution of benthic biofilm to arsenic biogeochemistry in freshwater environment, and precisely in the water benthic biofilm in interface. Lastly, this study show that the importance of biofilm as indicator of the quality of freshwater ecosystem. Thank you. That's all from us.